Welcome to the Conversational Flow, a KCAS bio podcast.
Hello, and welcome to the 26th episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry industry for over 14 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our headquarters are located in Kansas, with an additional location in Europe and a partner location in Australia, I'm here in Philadelphia, Brian is in Atlanta, and Jeremy, our producer, is in Missouri. So we are truly all over the map. We're amped up to have you listening to the 26th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing, before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're excited to have you here, and we're looking forward to a fun episode today. To kick us off for episode 26, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got a packed agenda again today. We'll start off with some medical research news and investment news. We've got some interesting, some KCAS news as well. And then I really like the paper that you picked out for this month's talk in terms of Basophil activation. So I'm excited to dive into that and the main topic as well. In terms of the medical research news, the first article I picked out was pig organ transplants and some of the work that a group is doing to find a way to stop immune rejection of those pig organ transplants.
Yeah, and I think this is really important. Some obvious parts, maybe some not so obvious parts, is one, access to organs, right? There's wait lists for patients who are in need, various types of organs, liver, kidney, so on. I know that there has been a lot of work in this field regarding skin transplants, so pig skin, not footballs, but pig skin there. And then just last year, the first pig kidney transplant was done by Harvard scientists. Now, the interesting thing about that was the first human organ transplant, which was a kidney, was done in 1954, and it was done in the same hospital. And so, that was pretty cool. The other part of the importance here is that, there is unequal access to transplants for ethnic minorities. And so having more options in the field certainly helps to even things out, making sure that everybody has equivalent access there.
That's exactly it, right? And, when we think about transplanting an organ in general, you basically have to wait until someone else is done with it, which isn't the easiest thing, of course, or you can try to make your own. If you try to make your own, that's a very, very difficult thing to fully manufacture yourself. So it would be best to start from some sort of scaffold or some sort of other donor as opposed to building one from scratch. Here we've got the thought of, can we instead use an animal and strip it of anything that would identify it as alien to the human body, which is the main advancement that they are describing in this article here.
Yeah, and we talk about rejection in the past before on a cellular level when we're talking about cell therapies, CAR therapies. We got to get that in here just about every podcast. In this case, what you're talking about, they're working on removing pig-specific carbohydrates, any endogenous retrovirus, things of that nature. And then they also have to add human genes for it to be accepted. And so things like complement inhibitors, anticoagulants, immune regulators. And so it's quite a bit of work to be able to generate such a platform.
That's it. But even with all of that work, it is still better than building it completely from scratch. And I see the utility here where you can think of either personalized issues where if you have a specific type of immune system, let's say HLA matching or something like that, then you could have a number of different genetic modifications targeted for that. And also you could think about this for other organs. They're doing this for liver because liver is a good sort of model system, but as you mentioned, it could be used for kidneys, potentially for hearts and other things. Again, pigs are excellent model systems because they do have similar size of organs to humans in many different cases.
Yeah, and so this one looked like it was short, so 10 days. There remains to be seen what sort of long-term viability could result from this. I didn't see any follow-up. Did you?
No, they did just publish the initial papers this past week, so I would expect follow-up in the next 1 to 2 years as well. This is implantation into basically brain dead donors, so not a walking human being that is using this as like a survival tool, but instead a person that may be in a coma or no longer have brain activity that has agreed to donate their body for science, and then can use this to understand if the livers are functioning and viable after implantation. So a very sort of contrived experimental setup that will tell them if there's an immediate and sizable immune response. But definitely the next step would be to incorporate this into a person that is a better demonstration of what a true acceptor would be for this type of organ donor.
Yeah, I did want to take a step back and mentioned about being 10 days. There are essentially two types of transplants where, it's the long-term, or at least the goal is to be long-term, and then the bridging transplants. And even if something like this were to be a bit shorter, it's not considered rest of life, let's say. A bridging transplant would allow for a patient to then be able to wait for at least have some sort of support before getting a human donor organ if that becomes available. As we mentioned right at the outset, there are wait lists for organs, and so it can take quite some time. So get something to get them to that point is certainly an important step.
That's it. With kidney transplants, you can remain on dialysis for some period of time, but for liver transplants, it can be a much more difficult process. Similar with other organs. So again, making these lists as short as possible is key to getting everybody the organs that they need and making sure that we basically save lives with this.
Yeah, so really interesting to see what other formats it comes into. As mentioned, we'd seen in the literature companies trying skin transplants and kidney, liver. It would be really a godsend to many patients out there.
That's it. And of course, the flow cytometry tie-in with all of this is that immune rejection is one of the key considerations for safe and effective transplant procedures. So all of this comes with a lot of different flow cytometry to understand how the body is reacting to these donor tissues and what the long-term efficacy may be of these types of therapies.
I was sitting here waiting to see how you were going to tie that in. That was very good. I like what you did there.
Yeah, indeed. And Yeah, the next article that I've got listed here, also from Nature News, so you can tell where I was searching for articles as we were getting ready for the podcast. But in this case, the article is around the godfather of AI. So basically, a researcher by the last name of Bengio has become the first person to hit 1 million verified citations.
And he was recognized among a team of computer scientists, including Jeffrey Hinton and Yan Li Kun, for the Turing Award in the past, which is awarded from the Association for Computing Machinery. It's specific for contributions of lasting and major technical importance to computer science. I'll take a tangent as kind of a video game nerd that there's a Turing test video game that I've played, really kind of a puzzle thing that I found a lot of fun. And so during the whole research of this, looked into Turing specifically, background there. It was very interesting. So highly recommend that there. But yeah, I mean, 1 million citations, that's incredible. Imagine to, like, within the field, it kind of, those citations grow exponentially after a while, because when you're one of the seminal authors, core authors, you got to reference where things came from. And even just for us, we've talked about this, the explosion of the use of AI, obviously monitored by our own driving task force, which you're a part of, has really paid huge dividends as far as efficiencies.
Absolutely. And, when we talk about a paper exploding, we usually talk about it exploding within the context of biotechnology, let's say, right? Like if you look at the trajectory of CRISPR citations, that in our eyes would be exploding. But this AI implementation, of course, exploded not only within the field of computer science, but across all fields almost simultaneously, which is where the trajectory of this citation list definitely ballooned. But I wanted to put it as a conspiracy theory that, maybe they put into their AI something that would propose citing their paper more often. And so that's built into the foundational structure of these large language models as we go forward here. It'll just constantly refer us back to that initial paper.
Do you think there's a limit there? I mean, I could see that as there being a paper that would follow that this is what. But no, I get that you're talking about a conspiracy theory. That's pretty funny to me. But hey, you never know. I'm curious about your experience as far as the steering committee for AI and our own use, and what's that been like, and what can we take from 1 million citations, exploding use of AI, and our own internal use?
That is a complicated topic, right? So absolutely, the use of AI is exploding in a lot of different ways. At KCAS, we take a very sort of risk-centric view of it. So as helpful as it can be, it is much more important that we maintain confidentiality of all of our clients and client information, and much more important that we do that than potentially get some of these one-time benefits. So most of the work of the AI Governance Committee is making sure that for any high or medium sensitivity data, and we do have a pretty robust classification scheme for how we classify data. But basically, that anything that's touching medium or high sensitivity data is absolutely walled off from any risk to clients, to client confidentiality, or anything else. So that is really the primary purpose of the AI Governance Committee, definitely to guide the implementation and make sure the implementation is effective as well, but primarily to make sure that everything stays where it's supposed to stay. So that way, if there is a foundational concern with this, and it keeps referencing the one paper, that it won't potentially interfere with any of the results or discussions that we're having with our sponsors.
And see, that's kind of why I wanted to delve into that a little bit with you. I know from my perspective, everything that we've done with AI has just been process driven and with an eye on efficiency. How can we do the same thing or better faster? I figured you were seeing even more than from what I've seen. There's always ideas on how we can improve things, but you're right. I mean, obviously, the security of data is tantamount, and so I want to make sure that we're uh, observing all the proper regulations and everything there. So appreciate that perspective.
Sure. Yeah, and speaking of AI and potential impact on business activities, definitely there is the investment news as well. So the first topic is, of course, Pfizer completing the acquisition of Metsera here.
And so there's a bit of a bidding war between Pfizer and Novo Nordisk for this acquisition. And you can understand then Metsera's lead candidate is a monthly injectable GLP-1 receptor agonist. So this is MET-097I, which is a phase 3 molecule. And do also have amylin analog. And so it's a kind of combination here when with amylin, you can inhibit glucagon secretion. So what you're looking for there is reducing glucose production, delaying gastric emptying, and then promoting safety. And so satiety, sorry, not I mean, obviously we want to be safe, but what that means is essentially the signaling fullness. And so when combined with these GLP-1 agents, have even greater efficacy than standalone. And so with that, part of the drive for this, I'm looking at the bidding war, essentially, is that their mid-stage trial data showed about 14.1% placebo-adjusted weight loss after 28 weeks. So very, very promising there, and expected to reach about 150 billion within a few years. The global weight loss drug market, that is, and having monthly injections instead of weekly, obviously increases adherence, right? We've talked about this before, that with the various treatments and therapies that are out there, they could do fantastic, but if you're asking the patient to do something more often than they're comfortable or whatnot, it just drives down the utility there. And so by having a monthly injection, it's much more favorable there.
That's it. And what I like most about all of these GLP-1 drugs and talking about the GLP-1 news is that it is basically putting drug development on speed run, right? Where usually with like an oncology drug, you would see basically a new drug targeting a specific mechanism of action, and then you would see maybe another drug from a competitor come in targeting the same mechanism of action, and they can compete based on how well they target or don't target that mechanism. So then there's that initial level of competition, and then you get additional entrants into the market that may target the entire patient experience and make sure, as you say, that the drug is better tolerated or more palatable to patients in one way or the other. But that process can take decades. Here, because of the popularity and the amount of money, basically flooding into this GLP-1 market, everything is happening so fast that we can see it in real time. Where initially there was just the GLP-1, and it was making one or two companies fantastically profitable, but then there was a competition on exactly whose drug was most effective, as indicated by the amount of weight that you could lose with it. And now we're already to the stage where people are competing in terms of how convenient that drug is to administer. And it's just impressive to see all of that happening in such a short time frame, because it is such a high impact to the balance statements, basically, to all these giant companies.
And having more options out there too does a wonderful thing in the market. It can tend to lower the costs of the therapies, right? We talked about previous podcasts that specifically these weight loss drugs can be very expensive. And so the more options you have out there, the lower the price will generally be.
Yeah, no, this is where capitalism is really working, ideally, right? Like, it can be a little bit inefficient that everybody's developing their own, but definitely I can see where this will eventually lead us to a good option that will be at a good price, and that's the end result of what we want here. So,
Mm-hmm.
yeah. And towards capitalism working here, the other story that I had for today is Merck entering into this R&D agreement with Blackstone, a private equity company, for the development of SAC-TMT. And I am going directly for the abbreviation there to neatly sidestep the need to pronounce that drug name.
Is it sacituzumab?
Sacituzumab tirumotecan. Yes, absolutely. SAC-TMT, we got it.
There you go. So,
Right?
yeah, Blackstone funds late stage assets and overseas trials. And so, interestingly, this deal here is to aid Merck in these assets and getting them to patients. Really, one of the things Blackstone is known for essentially aims for an 85% success rate in phase 3 to approval, which is very high, very successful, and so that's where this partnership is coming from.
Yeah, and basically these phase 3 trials are enormously expensive. So even for the giant pharma companies like Merck, running all of those phase 3 trials would have a pretty significant impact on their balance sheet if they were to finance all of that cost themselves. So we had a story on one of our previous podcasts about BMS basically spinning out a company to take on that risk and that R&D expenditure, as opposed to keeping that all on the BMS specific balance sheet. And this is just another example of that, where, again, this late stage development is being financed by a private equity company. But you can see the attraction for a private equity company as well, where, if it's already past the phase 1 and 2 trials, that takes a lot of the risks out of the investment overall. And then also, if the drug is being run by a sort of brand name like this, we'll call Merck, you don't have to worry about the potential variability of different management teams or different management styles that you may see at smaller biotechs, because all that is done by an engine that's done this many, many times successfully and has a lot of very robust processes to make sure it's done correctly. So there's a real benefit to invest this type of investment for private equity as well.
Right. And, at the risk of sounding like a show for Big Pharma, which, I've been in the industry for a long time, but certainly wanted to inform. Something like this demonstrates the high cost of getting drugs to market. And you were just talking about, there's failure rate too. The failure rate of drugs getting to market is fairly high. And so what ends up happening is there's a lost cost for those, but then the cost on top of it for these drugs that end up making it. I know a lot of times you'll hear people argue that it costs X amount of pennies to make this drug, or something like that. There's a lot more cost that goes into a drug than just the actual manufacturing of it, which in some of these we've talked about, the cars can be very costly for manufacture, but it's the clinical trials, it's the development, it's the failed trials for other therapies that just don't make it to the market, and that's where a lot of that cost can come from.
That's it. Yeah, so hopefully this is a good way where private equity capital can help speed drugs to market. Big pharma expertise in phase 3 trials and just scale can help drive this as efficiently as possible, and everybody can benefit from new therapies that get to market faster. So an interesting approach, and we'll see how it evolves over the next few years as private equity and different types of funds look for different ways to make sure they produce capital. So interesting
So
overall.
I know at the beginning of the year when we were talking, and this was right before the one where I actually made the agenda, and it was catastrophic for you for internal reasons. But we talked about mergers, acquisitions, and so you keep an eye on that even more so than I do. So, what is your thought on the direction there?
Mergers and acquisitions are, I would say, up for the year compared to 2024, but it has gotten a lot more lopsided, where there are significant winners and there are a lot smaller number of winners, but it is a lot higher dollar value, and that I think reflects two things at one time. That just the costs of doing any of these things has gotten higher. So the only amount of money that is meaningful is also high. Right. So if a trial used to cost 50 million, maybe now it costs 75 million. So now you need to increase the size of your investment if you wish to get it past the next milestone. So that's definitely a thing. But then also, I think the industry and capital in general is being more careful and more diligent with their investments. So they're picking the winner they want, and then they're spending the money it takes to accelerate those all the way to a longer or a more distant milestone than they were previously.
And do you see it continuing in that direction, or is this more driven currently by the current financial climate and when things ease up a little bit, thinking that that's going to go in a different direction?
I see this being a long-term trend. I'll just leave it there. I think that there was a spurt of activity around COVID, and a number of investors got into the market that were not the traditional biotech investors, or may not have had as sophisticated of screening algorithms and things like that. And I think that now that has largely exited the market, and we're back on like a steady state. And I think people have just adjusted to the increased cost of doing things, and that will be the norm going forward, if I had to guess.
No,
I
mean, obviously you're reading crystal ball, looking at finance, looking at the market as a whole. So a lot of nuance there, and certainly respect what you're putting together. I know, like I said, you're definitely paying attention pretty closely to that stuff.
That's it. Yeah. And in terms of where we get this information and who we talk to about it, definitely that comes a lot from conferences. That comes a lot from our peers across the industry, and of course KCAS. In terms of KCAS news, we did attend, of course, a MOSCA event here in November in the Philadelphia area, as well as SITC, the Society for Immunotherapy and Cancer, if I remember correctly.
Mm-hmm.
And also AAPS PharmSci 360. So all those we did in November are ready, and we are expecting to also attend EBF here in the next week or so. So that should be the end of conference season for the year, and a whirlwind it has been, definitely.
Yeah, you've been to a lot, you've done a lot of traveling. And so it starts up again, April, May timeframe?
Some as early as March, but yes, absolutely. There's definitely the fall season and the spring season there with most of these events.
And mentioned about travel. So have you been impacted by travel at all recently?
Oh, so badly. As you can probably tell from my microphone, I am not on my podcasting microphone today because that is in a different city. I am stuck in Atlanta at this point due to some canceled travel there. So apologies to listeners for sound quality for this episode.
See, sounds good to me. So I don't. Hopefully, no worries there. But I know you miss it. You travel more than just about anybody I know.
We're very good friends with Delta, so always good. But yeah, in terms of KCAS news, excited about all of the conference travel we had. And as you say, we're already planning out what we're going to present at conferences for the spring. So we have a lot of interesting internal projects. I think there's going to be a lot of standardization of spectral flow discussed next year. Hopefully we get some good industry consensus on best practices and next steps. So I'm looking forward to those talks and meetings.
Right.
But in terms of the paper of the month, you did select this one, and I really enjoyed this one actually. So let's maybe jump into that one.
Yeah, so the paper is flow-based Basophil Activation Test in immediate drug hypersensitivity. This is an EAA-CI task force position paper published 2024 in Allergy, which has an impact factor of 12 based on what they had on their site. Mayorga and team authors here. And so really looking at specifically the use of the Basophil activation test and trying to get to some sort of consensus there. It's a very important test. And, taking a step back, I like to do a little bit of look on history. And so go all the way back to 1879 and Paul Ehrlich identifying cells with dark blue granules that stained in peripheral blood. These were termed basophilic granulocytes. And then this Basophil activation test was actually discovered in 1991 by Edward Knoll and is important for both the diagnosis and monitoring of allergic diseases. As far as allergy, the term allergy was coined in 1905. And so this was specific to immune response in hypersensitivity reactions. And so what is important here? The Basophil activation test, or BAT, reduces the need for in-vivo procedures. So some of those are the intradermal challenges, allergen challenges. We don't have to rely on those that could potentially cause some kind of unpredictable severity. This is done on blood samples, and here you essentially challenge the blood with some sort of allergen, and then you process and stain with markers. Here specifically, we've got CCR3, which identifies basophils, and then CD63, which looks at the activation state. And there is another kit uses CD203 as the alternative to identifying CD203C, but still looking at CD63. Good thing is it's a fairly simple assay as far as flow assay is concerned. We're not talking about 45 markers, just talking about a handful of markers, and can be run on many instruments and looking at that change in CD63 to understand the levels of activation.
Yeah. And as you say, this is an incredibly important procedure, both for drug development overall, again, where we can predict and avoid these allergic drug reactions, basically before they get into patients. But it's also one of these super, I'll say in this case, simple tests where it's just a couple colors. So technically not very challenging just from the analysis standpoint, but the treatment procedure is incredibly important, and how you decide which version of the Basophil test that you're going to implement, definitely very, very important here.
And at this paper, they actually did a survey across Europe. So there were 106 respondents and identified that there's a large variation in protocols. Some of the different things that they saw differences: drug handling, the panels used, interpretation, the sample type. And so it makes it difficult to compare from lab to lab. And inhibits their large scale adoption.
That's it. And the reason that this really flagged to me as, oh, this is a really cool paper, is that it has at the end of it a table of consensus recommendations. And I know I'm jumping a little bit to the end of it, but I thought that this was the coolest thing that I had ever seen. One of my gripes with papers and publishing in general is the question of what value is the publisher adding to this, right? Like, you submit a paper somewhere, a volunteer screens it in some cases, or sometimes a professional editor, but someone screens it. They send it out to peer reviewers. These peer reviewers are generally not compensated, so these are just volunteers that are helping to do things at the goodness of their heart. And they review it, provide their comments. The author revises as needed. They publish it. There's either a publishing fee that you pay to get your paper published, or they charge users per usage, basically, of the paper to download it and understand what it's saying. So, in terms of the value of the publisher in that case, it's almost like a marketing value as opposed to a actual contribution to the field. With this table at the end, it provides a really interesting framework of, could you establish this type of table and have people always sort of contribute against that type of framework for building on academic or medical knowledge bases overall. And this paper is focused on a pretty specific area of medicine, so it makes sense to have this table, and this is the right group of experts to be able to contribute to it. But how nice would it be if each paper had their own sort of table of knowledge and what is known and not known, and then authors, as they're generating research topics, could plan on where their paper fits into that body of knowledge somehow.
Yeah, and it gives the recommendations so that you're not starting from scratch if you're coming into this area. You're a lab that needs to test this and you're not exactly sure the direction. Breaking it down even in smaller pieces is one of the things that we try to encourage. Is if somebody's got a problem when they're here at work, not only do you look at what is the problem, how can we solve it? And I think that's what you're saying this paper is offering, right? Is solutions, not just, hey, there's a problem here. With the BAT test, I think one of the interesting things that just in understanding the test and doing further research for specifically this podcast, understanding how it compares to other tests. Like, for example, just looking at levels of total IgE and understanding that doesn't always provide information to specific allergens. And then if you have specific IgE specific to an allergen, does not always equate to a clinically meaningful allergic response to that substance. And so the BAT test really is giving you some more insight here when you're adding the allergen. You also have to be careful, though, whether or not it's a peptide truncated form or you're talking about a full length form, and what you're using for activation there. But having the understanding of how to use this best really drives that field forward.
Absolutely. And you're right. Basically, this is being described as sort of a complementary test for some of these serum-based tests or soluble biomarker type tests. It's giving sort of a different view of information. As you can see from the paper, it's used almost all the time for some types of drugs, and then some of the time for other types of drugs based on the expected mechanism of action there as well. Definitely there are some cost implications to running the additional test, but anything that can be used to increase the specificity or sensitivity of this test, as described under the receiver operating curves, is critical towards screening out good drugs and making sure they get into the clinic, or stopping bad drugs before there's a severe adverse event potentially in the clinic there.
Yeah, and it's part of an entire package here too, right? Healthcare providers are going to look at the history, order skin test, then perhaps goes to the Basophil Activation Test, and then if that doesn't work, going to a drug challenge, of course, assuming that there is some safety there for the patient. And so, yeah, this definitely one of those tools. We talk about this a lot, the diagnostic toolbox and analysis toolbox, and how to properly pair things. And that's where this proposal from the paper really sticks out in guiding how that process should play out.
Sure. And they do call out some of the things that we've seen, because we also run the Basophil Activation Test, of course, at KCAS and a couple of our different labs. Some of the things they call out in the paper are some of those common things that we've seen, both with Basophil activation testing and other things where you think you know the patient's immune state at the time that you're testing them, but can you ever really, right? Like, is that person currently sick, not sick, allergic to something that they don't know about, taking some sort of medication, because as humans we have just a wide range of maladies, and maybe this medication interferes and maybe it doesn't. The number of questions that you might have to answer before getting a fully qualified result here is staggering, but can still be teased out as needed in the context of a clinical trial in some cases. We've that's why whenever we're running Basophil activation test validations, we'll always usually include at least 5 donors instead of 3, or in that ratio, to make sure that we can exclude anyone. Because I find that especially with basophil and basophil activation states, that's one of the most common items that people may not be aware if they are having a mild allergic reaction. And then you do get a much higher donor variability in the resting activation state with basophils.
It's always a complicated effort to analyze clinical data when you're talking about many different patients. Some disease states are easier than others. One of the classic ones that we always talk about is AML and the heterogeneous nature of that disease. But yeah, I mean, that's a great point.
Sure.
I did want to take a step back, and so we had called out at the beginning of this it was the EAACI, so that is the European Academy of Allergy and Clinical Immunology.
Okay, yeah. So I mean, the right group to be commenting on this, with European clinical trials in general, there's a lot more involvement from, I would say, medical faculty. And this group has gotten together and made recommendations. That way they can standardize between different groups and make sure that moving forward everybody's using roughly the same procedure and roughly the same rationale of when to apply it and when not to as well.
Well, and that's a good point. Something I did want to highlight in talking with team members who are running this day in and day out. There really are only 2 commercial approved kits, IVSE kits, and they're not aligned in terms of the markers. Now, I'd mentioned this at the outset that they both use CD63 as the activation state, but they don't use the same marker to identify basophils, and so it could lead to a little bit of a difference in interpretation there. And so, again, with the recommendations, trying to get to a standardization.
Sure. Yeah, and that's what we work towards on a global basis, on a regular cadence here. And glad that medical institutions are doing the same. It can only improve things. Yeah. And any other, I guess, sort of consensus recommendations or things that you really wanted to call out in terms of how to process the Basophil activation test? Or
I would actually take a step back and just go more general, right? This is where the industry continues to move. And when I talk about the industry, the pharmaceutical industry, clinical testing, flow cytometry, there's so many tools out there, and I think we've got a gigantic wealth of information from so many clinical trials, so many years, so many patients, all different disease states, and you can't take all of it together because of the lack of standardization. And just imagine if there were more standardization, just how much more information we would have on disease states and treatments that we just don't right now. And that's where it becomes important to get this sort of standardization, not just for the Basophil Activation Test, but in so many other areas of science. And I think we can only stand to benefit. And so that's where this was not only interesting to me for this specific test, but in general, because we're talking about, spectral flow cytometry, and what do we use for reference controls, and how do we analyze the data? How do we identify populations? All of these things, the scientists have very good ideas and really work towards getting the best data possible. But there are more than one option to be able to get to an end result.
Yeah, and when I think about standardization like that, right, I think that it starts with this type of, here are the general process requirements to get things done as like a first step. Maybe the second step is some type of standalone automation that is geared towards implementing this procedure in a standardized way. And then maybe a third step is a specific piece of equipment that is geared towards doing this on a regular basis in some sort of IVDR or PMA compliant manner. So like a medical device, if you will. And I'm curious where you see the level of standardization needed to really apply this across the drug industry. Is it at the automation stage, or is it at that fully compliant device stage?
And you're talking specifically for this test, or
it can be a general question about the level of test, but we can think about it in the context of this test as well.
I think that the IVD platform drives more standardization outside even of that one area, just because the regulations that go into it to get to best practices. And obviously, when you're looking to develop and new panels, new assays, you want to use best practices. And so while I don't know that the entire industry needs to go that direction, certainly I think it's a great benefit for the industry as a whole. Automation too. We always talk about taking out the human element. I mean, you don't never want to do that completely. We'll always talk about specifically gating, right? Analysis of data, and do have automated tools that help us to get that done more quickly. But we always make sure that scientists with experience in the area have their eyes on that data. And so automation is certainly necessary, but it's a part of the picture, if that makes sense.
Sure. Yeah. So then, if the pathway is agree on roughly how it should be run as step A, then make a robot that can do it really fast and really precisely as step 2, and then make an IVD kit that everybody uses as step 3. Step 3 is really the one that has the widest benefits for comparing results from different trials. But definitely you need step 1 and step 2 before you get to the level of a step 3 there. So, yeah. All right. Interesting. Anything else that you wanted to bring up about the paper overall, or should we move on to, I guess, the next part, which is banter?
Yeah, I think we did a good job there. I again really enjoyed the paper, and it sparks a lot of discussion. I'm sure we could continue to discuss for days on that. So why don't we go to next steps here?
Yeah. So next up is fan feedback, and in line with our paper, of course, we want to understand from our guests, what are you literally or figuratively allergic to? Some people have dust mites. Anyone that has gotten bit by the wrong kind of tick may now be allergic to red meats. So interested in the range of allergic allergens that our guests are concerned with.
Can we include stuff like cranky people, bad drivers, things of that nature? Or does it have to be
Well,
more?
that's where the figuratively comes in, right? So, like,
I was
no,
just thinking
like
about driving into work today, and it's almost every day run into somebody that I think I must have an allergy to, because I do get a reaction pretty quickly.
that's it. That's why we ban you from the blood draws related to Basophil Activation validation, Adam.
That's a good one. I like
Yeah.
that. Yeah. How about you? Any? I guess you're at home, so you didn't have any allergic reactions on your commute today. Anything like that for you?
Not on the commute, just snow. I'm going to say snow is my primary figurative allergy here.
So we can't have you doing any blood draws during the winter months, is what you're
That's
saying?
it. Spring and summer only.
It's funny. Okay, I'm going to go on a total tangent here, and I want to put this out. It's not flow cytometry. Like you mentioned seasons, and so my daughter and I were having a conversation. I had recently gotten a new cologne, right? And colognes, I didn't realize. Like, I'm not big into this industry or whatnot, but they're marketed for different seasons. And the one that I got in the literature for it, it said it's ideal for fall, winter, and spring. Now, I will ask you, does anything about that strike you as odd? Because it didn't strike me as odd, but I will give you more after you answer that.
It's a little weird because I would group fall and winter together, maybe, but not so much spring.
Okay, so this is the answer that everyone's giving me, and like, whatever, it's a smell. Like, I don't know how much it matters. What? Yes, that was exactly my daughter's mention. Was how can it be good in spring and winter? Those are two completely different seasons. One's generally warmer, one's cooler. Like, I could see fall and winter, but not. It's like. So, anyway, I digress there. Evidently, there are very specific seasons for certain colognes and certain smells. And as we were getting in the car, she goes, "Oh, you got your cologne on." She's like, "I swear I smell vanilla and caramel and this and that." I go, "Do you really smell like what did I do? Did I take the chocolate salted caramel things that I got and I just lathered it up? Like, is that like what? I don't smell these. Like, I don't know. I know it smells nice. That's the extent of my knowledge in colognes here.
There is a whole industry, Adam, about creating new scents, testing scents. So yes, it is a huge area. Very important for some people. Other people may not have the, I don't know, palate. I don't know if that's the right word, but the ability to smell all of the different variations that may be in there. But
What?
yeah, very interesting overall.
We've talked too about bourbon, right? Bourbon
Yeah,
tasting, and there's notes of vanilla and whatever in there. And it's another one where I don't know that I necessarily get those nuances. I don't think I've ever tasted vanilla in a bourbon. So, what are your
I
thoughts? Do you taste that?
I can taste vanilla and different notes of like oak and things like that in bourbon. I worry though, Adam, that you are jeopardizing your chances of being a spokesman for Angel's Envy at this point because if you can't differentiate the different bourbons and things like that, they might withdraw their offers there. But
I can tell between a good bourbon and a bad bourbon. I'm just not going to be able to tell you what notes there are primarily in there, if that's fair. I think
okay,
don't you want the everyman or the everywoman, whatever, to be able to tell this is a good bourbon, right?
yeah,
You don't need
that's
to
it.
be a bourbon snob to be able to tell what's good and what's not.
Yeah, the marketing slogan. Even I can tell that this is better. So,
Exactly. Even I, yes. It's a good one. All right,
yeah.
so let's before I completely drown this podcast in other things, what do we have next?
No, that's it. So next, we're just going to highlight that for our next episode that will be in December, and we'll be talking about holiday lights and flow cytometry, and really thanking our listeners.
Yeah, yeah. Here, last question then. Do you have a cologne?
I do not, Adam.
No cologne?
No, I mean, I just run around. I try to shower regularly. I don't know, maybe some deodorant, but
That's awesome. All right. So like you wouldn't notice the different notes or the seasons for the different smells and all that kind of stuff either.
That is not me. No.
I do enjoy it. I'll be honest. I mean, I had cologne since I was back in high school, and I just had this memory of my brother coming down, and he stunk up the whole house with his cologne. He must have put about 25 sprays on in one time, and he went to a school dance, and that house, like our house, smelled like his cologne for a long time after he was gone.
That's it. I think about my experiences with like Axe body spray and things like that in high school, and I'm just like, I don't need any of that. And then, of course, in labs, we do recommend against strong smelling scents and things like that could potentially impact experiments with various volatile chemicals. So there's always that I can lean back on. But
yeah, I do pay attention to that. Make sure if I pick something, it's not an overtly powerful scent, and or not wearing something. But anyway, yeah. So I'm done.
All right. Well, thank you again to all of our listeners. I've greatly enjoyed the episode. Looking forward to tuning in for the next one in December here.
Yeah. Thank you, everyone. Take care.